Thank you very much. So I will go through Covaris, what we have in terms of workflow solutions for multi-omics. So we can say any kind of omics, and I will show a little bit why that is. So the basics of everything we do is on the adaptive focused acoustics technology, so AFA. So if I say AFA from now, it means this acoustic technique they have. And if you look at the slide, do I have a pointer here? Yeah. So if you look at the little slide on there, you see that it's a tube with a sample inside. And what we can do with the technology is to focus all the energy from an ultrasonicated generator into the tube. So the tube is in no contact with its surrounding. It's in a closed tube, so no cross-contamination. It's also temperature controlled in a water bath, so it's an isothermal process. And we can control the energy input to the sample in a very accurate way. And we vary that depending on the application. So what you probably know most about Covaris is the shearing company, the DNA shearing company for library preparation. And you probably recognize these two instruments that have been around for many years in a lot of labs. But that is not where we are today. So we are really transforming. We are since a few years back in the transformation of going into multi-omic sample preparation. So this is two examples then from genomics, where we have the germline workflow is much more than shearing DNA. We have for sample collection solutions, we have for whole genome sequencing, exome sequencing, library preparation for germline, also for somatic solutions then for extraction from FFP. And I will go into the extraction and the collection in two examples here. You see at the bottom to the left there a new kit we have just launched for blood collection and stabilization. But I'll show you a little bit about that later on. But the integration of sample handling, sample preparation in any omic solution is what I want you to take from this session here. So we can more or less prepare biological molecules from any biological sample, and we serve the market then with any kind of market that use biomolecules. And that's what I would like to show here, that we have instruments from single sample handling up to high throughput. We can work in 384 plates if necessary. We have the consumables and reagents that are optimized for the application we have developed, and we can automate this. We have a dedicated automation team. We have a formal collaboration with Hamilton, but we can put our instruments inside a system or outside a system with any liquid handler. But we develop most things together with Hamilton. We service any kind of market using biomolecules, and the supported application range from all kinds of omics. So we have solutions for genomics, transcriptomics, epigenomics, proteomics, and also metabolomics. And we have examples of customers that have optimized their workflows. On this aspect, I also would say that the other year we were accredited according to ISO standard, allowing us to develop IVDR solutions. So in the autumn, we will start to launch both instruments and reagents for CE IVDR. And we plan to be compliant with all instruments and kits that are of relevance for clinical use. And on that aspect, I just want to start by showing TrueCover, which is a range of kits we launched during the last year for library preparation. And we have for whole genome sequencing PCR-free and an amplification module together with this. We also have a total RNA library kit to which we have, well, that includes then rRNA depletion. But we have also now a module for globin depletion that comes up with this. And the last example here, we will come with more TrueCover solutions, but we also presented a few days ago, we presented at the ESHG about the Illumina 5-base workflow, where we have a collaboration. And I'll show a little bit about that as well for mutation and methylation combined analysis. I would like to start to show our solution then for FFP extraction, which we call TrueExtract. So this is quite innovative and different from any other vendors on the market. See if I can point out here. Yeah. So we use the AFA technology to emulsify paraffin. So that means that the tissue pieces we have will be get in very small entities and go out in an aqueous buffer. And in this way, the smaller pieces we have, the more efficient the downstream protein K treatment is. So we don't need to use any organic solvents for this. So in terms of efficient extraction, we can have very good quality of both DNA and RNA. It's a simple workflow where you centrifuge and you get in the pellet, you get the DNA, and in the supernatant, you get the RNA. So DNA and RNA from the same sample is extracted. And downstream of this, we can do any kind of multi-omic analysis, and this can be handled then from single sample up to very high throughput. And we have examples. So down at the left is one of these Hamilton systems, which there. It's a star. It's called the sonication star, an assay-ready workstation where the FFP extraction is handled completely automated with a decapper, capper, and our system is handled outside the robot. But this is developing in collaboration with LabCorp and Hamilton, and is used in high throughput for comprehensive genomic profiling, the FFP extraction on upstream of that, but also for MRD assays and any kind of diagnostic assays as well. And just to show some brief examples of this was a validation project from a US service provider. And you can see just evaluation of DNA based on qPCR. It shows that the delta CT values or delta CQ values for Covaris is considerably lower than the CT values of the market leader that was compared with and was formerly used in this lab. Below there is an RNA just measuring the amount of RNA, and you can see that the amount of RNA coming out from our solution is considerably more than from the competitive solution. But more striking, DNA is fine, but RNA sticks out, and you can see the evaluation to the right where the same samples have been extracted with a market leader and compared with Covaris. And the red dotted line there is the DV200 score that they require for the QC. And you can see all of the samples are okay from Covaris, but quite low for the competitive solution. And striking also, if you look at the size distribution here to the right, you see the black lines here, that is from the market leader, and the Covaris solution is the blue line, and it's a considerable difference between the size of the extractions. So RNA really sticks out as much higher quality. This was in a project, as I said, this was developed then in a collaboration between Covaris, LabCorp, and Hamilton. And you see the platform that was developed then on the slide there. But I think more interesting is that LabCorp evaluated after the implementation. This is I should explain it. These bars, the green ones are where they can do full reports, and it's the monthly reporting statistics. And you can see when they adopted our solution, when they adopted the Covaris solution, you see that the top part of the bars, the failed frequency went down drastically. And what they calculated is that LabCorp could save $1.3 million per year just on one of these setups for 10,000 samples per year. And the main savings of this is that they didn't fail with library preparations. If we connect this then, so I mentioned that we have library preparation, we call it the true cover whole genome sequencing. And there we, as I said, we have PCR-free, where you can go down to 50 nanogram input without any amplification. We can go down to 0.1 nanogram or 100 picograms then for with an amplification workflow. And if you combine this with our true extract, we say we can go down to 1 nanogram with an amplification workflow for FFP DNA and have very good results. In connection with this, also the TrueCover Total RNA is also compatible with the FFP sample coming out from our TrueExtract kit. And here we have kits where we can go in with, officially it's 10 nanogram and up to 500 nanograms, but we've shown lately that with some modifications we can go down to 1 nanogram input of FFP DNA or RNA. And I want to emphasize a little bit about the RNA workflow. That we have simplified a lot of steps here. So we have single-step rRNA depletion, and also we just combine this then with globin depletion. We have one single-step cDNA synthesis, and then all indexing is done in a PCR step. So everything is simplified, and the total time for RNA library preparation is 4 hours, at least 1 hour shorter than anyone else on the market. And again, we have the TrueExtract. We can connect this and optimize the complete workflow. So with TrueExtract, we connect the DNA and the RNA with our library preparation. And we have optimized this also for FFP samples. It can be used for any sample, but we have dedicated workflows optimized for FFP. Deborah will talk about the PacBio. I just showed this is a poster from AACR a few weeks ago, and she will describe then how PacBio has shown that DNA and RNA from the FFP extraction can be used for long-read sequencing with the PacBio library preparation. But she will discuss that. I also want to emphasize the Illumina Phi base. We have had a collaboration, or we have a collaboration to develop workflows for Illumina. So the traditional way of methylation sequencing with bisulfite sequencing means that you convert unmethylated cytosines. But the new version then, the Phi base, in the chemistry there, you modify, you convert the methylated cytosines instead, which means that you conserve the complexity of the genome, which means you can also do mutation detection and methylation detection on the same run. And here with the collaboration rendered that we have in our workflow where cells, fresh frozen material can be used, and we share the DNA with our devices, but lately also we optimize the workflow for FFP and methylation analysis. So we share the DNA, and that the DNA that is prepared either from fresh frozen or FFP goes then into the library preparation with a Phi base workflow. One more thing I want to emphasize about what we have launched recently is the TrueCollect. So this is a whole blood collection solution. It's a finger pricking device that you get blood on your finger, you put a small capillary device taking in 50 microliters from this. This goes into a matrix that stabilizes the blood in a tube. This dries out, and it can be sent by regular post, or it can be kept room temperature. So we tested this in a different ways to check the stabilization, and it works fine. So the sample is sent then to a centralized laboratory, DNA is extracted, and you get very high quality DNA out of this. So in an average, I'll show you what it comes out, 1.5 microgram of DNA at the range of 40 kb. Just briefly about the verification study that was done. We could show then, as you can see on the DNA quality, we have about 40 kilobases in the range of the size range. DNA quantity around 1500 nanograms. This is the storage where we have storage in over time. So it's 3 and 6 months. No, it's up to 3 months, but we have tested now to 6 months, and it's still stable after 6 months. Temperature stability, we tested up to 75 degrees, I think it says. My glasses are not good enough here, but I think it's 75 degrees. And it's no problem to keep the stability of the DNA. We think that this can be really interesting if you just look at any decentralized trials or diagnostics where remote patients are taking their own samples and send in the samples. For example, newborn screening or rare disease diagnostics. It could also be for population studies where geographic samples from a big geographic area is taken in. So we hope this can raise a lot of interest, and we will test this with customers now. We have a verification showing very good data from this. It's like you have the kit which is sent to the patient, and they take their own sample then and send it in, and in the central laboratory we have a kit then for rehydration and extraction. And as I said, very high quality of this. And we have it now for DNA. We know it works for protein. So protein will come in the autumn kit for the same sample, can extract DNA and protein, and we hope that RNA, that we can stabilize it enough to also extract RNA from the same sample. But that will come later on. But if you're interested in this, just let us know. Last, I want to just also to show that we have a solution for proteomics or any kind of omics. But for proteomics, we have dedicated workflows which are for the sample preparation upstream of mass spectrometry or any other kind of protein analysis. And here we have also dedicated workflows where the same instrumentation is used for proteomics work, where you can extract, you can disrupt and lyse tissue, you can homogenize it, purify it, and then we also use the instrument for enhanced digestion in a mass spectrometry workflow. So we can shorten the digestion from overnight to 1 hour by using the alpha technology. And we have some kits for this as well. So we have the protocols for fresh frozen. We have kits for protein FFP extraction and also from mammalian cells, and the full workflows for the trypsin digestion and also loading directly onto mass spectrometry. So what I would like to emphasize is that we are not only a sharing company. You can use our devices and our reagents for a lot of purposes. So we really think we can call ourselves a multi-omics company now with solutions for any kind of omics solutions. And we can automate this as well.